Hey everyone, today we're going to get into the first of our anabolic processes. Instead of breakdown and catabolic processes, we're going to be looking at the biosynthesis of carbohydrates. So, one thing to note here, we've been talking a lot about animals and how they break down energy products, things like lipids, amino acids, and especially sugars, and then make them into energy via ATP in particular. For sugars, we have the means to break down sugar into carbon dioxide and then via respiration make that breakdown products NADH, FADH into ATP. Now, we also can convert some sugar precursors into sugars, but we don't actually have the means to build complete sugars from base carbon or just from CO2. So we can do things like take pyruvate, which is a precursor, and turn it back into glucose, or we can take amino acids and also convert them into precursors to eventually turn back into glucose. We do have the means for that. And we talked about this in things like the pentose phosphate pathway, or in our amino acid chapter, where we looked at amino acids and the pathways they go through for energy transfer. But we, again, cannot just take CO2 and build sugars. So today's chapter, it is the biosynthesis of sugar, but we're actually going to be shifting gears and looking at other organisms, mainly plants and prokaryotes, and their ability to actually take base carbon and CO2 and build up sugars anabolically. So one thing to note when talking about the processes of building sugars is a difference between two pathways, oxidative phosphorylation and photophosphorylation. So oxidative phosphorylation, we just went through. This is the means to get electrons from cofactors like NADH, FADH2. They're then passed through a chain of enzymes, the respiratory chain or the electron transport chain, with oxygen as an electron acceptor at the end. This is why we as animals need to breathe in oxygen. And that energy is then used to fuel the production of ATP through ATP synthesis. And again, that's by making a gradient of protons. This is what animals use as a means to take electron energy and make it into another product of energy that we can utilize in our biological organism. Photophosphorylation is a similar process. In this case, it's kind of the reverse. Water is a source of electrons, and it is then, these electrons are then passed through a chain of proton transporters, with the ultimate acceptor being NADP+. So again, the reverse. Oxidative phosphorylation, NADH, dumps off electrons. Those electrons go to oxygen as the acceptor, and the oxygen makes water. In photophosphorylation, we're taking water and we're going in reverse, passing electrons ultimately to a carrier, NADP+. The thing that fuels this gradient of electrons is light energy in the case of photophosphorylation. It is used to separate charges on chlorophyll to eventually generate NADPH, and also to phosphorylate ATP, so to also make ATP. Here, oxygen is a byproduct in photophosphorylation because we're going in reverse. We're taking the electrons out of water, making oxygen. Photophosphorylation, again, is a light-dependent reaction that organisms can use. And it's a reaction of photosynthesis that generates, again, NADPH. I want to be clear, it is NADPH. We've seen this already in animals. We also use NADPH as animals, but plants use NADPH, and other organisms like bacteria will use NADPH as its main carrier molecule. So again, photophosphorylation generates NADPH and it generates ATP. And these are then used for a separate pathway in which they are used as energy to make structures and make sugars. So these are complex compounds like glucose and sucrose. Again, I want to clarify that there are two separate processes happening. Photophosphorylation is the actual process similar to electron transport chain that uses light, generates energy molecules. It generates ATP and it generates NADPH. These are then used in other pathway reactions, carbon assimilation reactions. Again, these pathways are unique to plants and other organisms. And those reactions are going to take the NADPH and the ATP we generated from light and turn it into complex structures like sugars. So let's talk a little bit more about this first step, the photophosphorylation, in which light is actually converted into ATP and NADPH in plants. This is going to occur in plant chloroplasts, and you'll see that plant chloroplasts have a very similar setup to our mitochondria. In fact, they are almost identical in many of the ways that they act. The inner portion of the chloroplast, these inner areas that we would call the mitochondria, are called cycloids, the cycloid. And here you can see the inner cycloid. Here's the cycloid membrane. And what happens in the cycloid of the chloroplast is that light comes in, and in its first step of the electron carrier chain of the cycloid, now don't get confused, this is similar to the electron transport chain. Different proteins, different mechanisms, but very, very strong similarities. Light is essentially converted and H2O, water, is taken and made into oxygen. So first step, light comes in, energetically releases electrons from water, which makes oxygen. So this is where the oxygen comes from plants. Plants we know produce oxygen. This is how. Light comes in, takes water, gets it to release electrons, that produces oxygen. Those electrons go through this electron carrier chain, and a very similar process happens. It's going to pump protons. In this case, we're actually pumping in the opposite direction. We're pumping protons into the cycloid membrane from the outside. So from the outer chloroplast area to the inner cycloid, we're going to be pumping protons in. So again, this is the reversal of what we see in the electron transport chain in mitochondria. This is going to do the same thing, create a gradient, and that will create a positive atmosphere on the inside with a negative charge on the outside. These electrons, again, their main goal is to pump these protons. And in the end, they're going to reach the end of the electron transport chain. and get put onto a electron acceptor, NADP+. This is going to make an NADPH. Again, it's the exact opposite, right? Instead of electrons being dumped off by an NADH, they are going to be accepted at the very end by an NADP+, and it's going to make that NADPH. Once all of these protons have been pumped and we've made an electrochemical gradient on the inside of our cycloid, that can be used with our good friend ATP synthase. So ATP synthase is the same ATP synthase, very similar to what we have. Notice that it's just facing the reverse way, so that in this way, the protons can pump from the inside of that cycloid out through the ATP synthase to the outside, where it will make ATP. But generally, the mechanism is the same. You can see that the photosynthetic machinery, the actual protons that are, or excuse me, the actual proteins that are part of the transport chain, this electron transport complex within the cycloid membrane, are very similar to what we see in animals as well. In this case, we have a system of 3 main proteins, and then, of course, our final ATP synthase, which is essentially the same. The 3 protein complexes that enact in plants are called photosystem 2, cytochrome B6 complex. This is similar to our cytochrome C complex in animals. And then photosystem 1. You can see that we're actually starting from photosystem 2 in the area where we are taking in light, moving through cytochrome B6, and then photosystem 1 is where you actually are transferring those electrons to a final NADP+ in the end. So we're actually going this direction, and eventually those protons that are pumped will go through the ATP synthase to generate our ATP. One thing that is a little bit different in this sense, I want you to notice that most of what's happening when we talk about the electron transport chain and the mitochondria happens inside the mitochondria. Everything's contained in there. Here we actually have many of the actions and the products happening on the stroma side, or the outside of that cycloid, including the ATP synthase, which is facing the other way, where we're actually producing ATP out into the outside. And again, this has to do with the fact that our gradient is reversed, right? We have a reverse gradient. It's happening inside and moving out. And so most of what's happening as far as functioning is happening on the outside in plants. So I want to do a quick overview of each of these structures. Again, they're very similar. They're very analogous to structures we saw in the previous chapter when looking at electron transport chain. Photosystem 2 is the area in which we are starting the chain of electrons. This is where light energy actually is coming in, and the light energy is then converted into electron energy. We're going to take light and break apart water into oxygen and protons, as you can see here. And via this breaking, we're going to take electrons from that water, and those are going to be the electrons that move through the rest of the complex. Now, within this photosystem 2, you can see one of the areas that's very important right here is this manganese calcium oxide complex. This is the complex that's actually responsible for capturing light and catalyzing that reaction into this breaking of water and forming electrons, right? Essentially electricity. This complex has been well studied, especially in the area of photosynthesis generation. If we want to get solar power, we have to do essentially the exact same reaction. We take water and split it, making oxygen and hydrogen. And during this split, we capture electrons and then can power electricity. So this manganese complex has been studied as a means to catalyze the capture of light. Once that occurs, the electrons are going to move through the complex through a special pair of chlorophyll molecules. You can see chlorophyll molecules are very similar in structure to heme. They actually have the same base structure. These electrons are then going to go to a carrier called a plastiquinone. Plastiquinones, which is abbreviated here as this PQ, plastiquinones are very functionally similar to ubiquinones. And remember, ubiquinones are also called this coenzyme Q. Remember, we saw coenzyme Q? It's a carrier molecule in the electron transport chain. Well, plastiquinones or PQs. PQs are very similar in their structure. They're lipid soluble. They can move through the lipid membrane and they can carry 2 electrons. The electrons from the splitting of water will get transferred to this plastiquinone in a plant, and the plastiquinone is going to carry it to our next complex, which is the cytochrome B6F complex. The cytochrome complex in plants is analogous to our cytochrome C complex or complex 3, which is in animals. Here, the plastiquinone is going to be received, and there's going to be movement of protons into the inner cycloid side, right? We're doing the opposite in this case. So plastiquinone is received. We're going to pump some protons inside the cycloid. And then the electrons are funneled to another molecule called a plastocyanin. These are analogous to our cytochrome C molecules, which, if you remember, were the carrier that we saw in the electron transport chain at cytochrome C. These plastocyanins can only carry 1 electron at a time, and they go through this Q cycle, same as the Q cycle that we saw in the electron transport chain. This is going to then transfer to our last complex, photosystem 1. Photosystem 1 is going to take in plastocyanin, and through a movement of electrons, and specifically through the use of a molecule called ferredoxin, it will then ultimately move both electrons to a NADP plus and generate an NADPH. This also is going to pump protons as well. Ferredoxin is the actual molecule which donates the electrons to our NADP, which is not shown on the slide here. But it is one of the main other molecules that traps electrons. Ferredoxin is used to trap electrons and then transfer it. Ferredoxin is similar in its nature to another molecule we've seen which helps trap electrons, which is this glutathione. you remember, we looked at glutathione, which is an energy used for in our regulation of reductive oxygen species. So one thing to note is that light-induced reactions are going to cause acidification of the internal lumen. The lumen is the inside of the cycloid in a plant. This is in contrast to what we saw in oxidative phosphorylation. Remember, protons are pumped into the lumen in plants, building up a huge gradient quickly, and it's in a much smaller space than in a mitochondria. And then this gradient is what's going to actually drive the synthesis of ATP, just the same as we saw in mitochondria through ATP synthase. The ATP has the same cost that we saw in animals, which is about 3 protons. It's really 3.33, but about 3 protons that are pumped back through ATP synthase are going to make 1 ATP. So again, here is that outline of all of the complexes. We can see this is where light is going to come in at photosystem 2. Photosystem 2 is going to transfer the light energy into breaking apart water to form oxygen and some protons. During this process, we also are going to get electrons, our energy currency. Electrons are going to move to a plastiquinone, which gets carried on to our second complex, cytochrome B6 complex. That's going to move electrons similar to our complex 3 in electron transport chain using a plastocyanin. Plastocyanin goes on to photosystem 1, where again, electrons are transferred through, eventually to the point where we have 2 electrons that are carried by ferredoxin. And ferredoxin is ultimately going to be what transfers the electrons onto an NADP+, generating an NADPH as one of our products. All of the protons that are pumped inside the lumen during this process are then funneled back out through ATP synthase from our gradient generating ATP. So now let's talk about, we talked about how plants differ from animals in how they generate energy products. Now let's talk about the actual anabolic process. Again, we mentioned that most of our chapters were focused on catabolism and how to extract energy from biomolecules. We're going to look now at how we do anabolic processes. In this case, we're going to look at how plants do an anabolic process building biomolecules. So again, plants can actually build molecules from CO2. And they can move intermediates between cells, just like in animals. We just saw that they can use sunlight to actually build energy molecules that they can then use to support this biosynthesis. So how do plants build molecules from CO2? The actual assimilation of carbon dioxide by plants is via these 3 carbon intermediates. So one of the most important precursors that plants will build is glyceraldehyde 3-phosphate. Remember, this is a precursor we have seen in glycolysis at the very beginning. Glyceraldehyde 3-phosphate and other triose phosphates. All this means triose just means 3 carbon phosphates. So glyceraldehyde 3-phosphate and other similar 3 carbon phosphates, triose phosphates, can then be used to build up backwards into making larger sugars like glucose, hexoses. These can then even be further utilized to build even bigger structures like sucrose, starch, which is a large complex carbohydrate, or cellulose, which is a carbohydrate that the plant uses for its cell wall. Plants can also use pentose phosphates, things like ribose, ribulose, and these can be used to build DNA, RNA, protein, lipids. So many of these processes we've actually seen analogous in animals. Taking glyceraldehyde 3 phosphate and building glucose, we already know how to do, right? That's gluconeogenesis, the reverse of glycolysis. Taking glucose and making pentose phosphates, that's the pentose phosphate pathway, which we've already seen. Some of these pathways are unique to plants, especially obviously taking CO2 and making the glyceraldehyde 3 phosphate is one that is plant specific. Making sucrose and making cellulose are also plant specific. Now, these processes I just talked about, things like gluconeogenesis and glycolysis and pentose phosphate pathway that we saw are ways that plants can get to producing sugar and producing ribose and other intermediates. These pathways that we find in animals, glycolysis and gluconeogenesis, are identical in plants. Plants can use the same pathways. Evolutionarily, we have evolved these pathways together. Again, the unique part is that they can take CO2 and make it into glyceraldehyde 3 phosphate and other triose phosphates. And then these can go into pathways that are identical in animals to then further build up larger structures. The area of plants that actually takes in CO2 and then does this process of assimilation to build our molecules occurs in areas called plastids. Plastids are organelles in plants and they're also found in algae. This includes things like chloroplasts, which are a type of plastid that's used for photosynthesis, and we just looked at that. And amyoplasts, this is a type of plastid that is used to just store starch. So, for example, potatoes have a lot of amyoplasts. They store a lot of starch in their cells, and you can actually visualize starch when it's stained with iodine that is filled up in these amyoplasts of plant cells. They have an enclosed double membrane, similar to mitochondria, and that inner membrane is impermeable to ions and polar molecules, again, similar to mitochondria. Similar to mitochondria, these specialized plastids also have their own small genome that is unique from the nuclear genome of the plant. So we have more in common with plants than you might have thought. Again, there are different types of plastids. I want to clarify, these are organelles. They're a part of the cell, a part of the plant cell. Chloroplasts are a type of plastid. Again, amyoplasts, which are storage plastids. There are chromoplasts, which actually just give pigment. They store pigment. These are going to be found in flowers, for example, where you have a very pigmented part of the plant. Elanoplasts are areas that store lipids. So plants that are very lipid rich or have high fat content are going to have a lot of these types of plastids. And then there's also proteoplasts, which are for protein storage. Actual CO2 assimilation does occur in a plastid. Specifically, it's going to occur in chloroplasts. So we just looked at chloroplasts and how they are used in this photosynthesis process. They also are going to be the site of CO2 assimilation. This is going to be similar to the reason that our energy breakdown pathways like citric acid cycle and beta oxidation take place inside the mitochondria right next to the electron transport chain. We want to have direct funneling of these products to each other. The area in the cell, the chloroplasts, again, is going to be the area where we generate ATP and we're generating NADPH. And it also is going to be host to what's called the Calvin cycle. The Calvin cycle is this pathway unique to plants in which we are actually generating our products from CO2. There is an intermediate in the Calvin cycle, just like in the citric acid cycle where oxaloacetate cycles through repeatedly. The Calvin cycle uses an intermediate, ribulose 1,5-bisphosphate. It's constantly regenerated using energy of ATP. The actual product of the cycle, though, is our glyceraldehyde 3-phosphate, our G3P. Remember that just like in animals, glyceraldehyde 3-phosphate can be in equilibrium with its isomer dihydroxyacetone phosphate. Remember, these are also formed in glycolysis. We see both of them formed and they can be interconverted into each other. They are isomers of each other. But again, we have ribulose 1,5-bisphosphate. This is our intermediate that is constantly cycling through the Calvin cycle. And we actually produce our glyceraldehyde 3-phosphate. This is the sugar product that we're going to make, precursor product that can then go on to go into gluconeogenesis and make actual glucose or whatever sugar is needed. The net result is that we are going to reduce CO2 and use an NADPH, that NADPH we just generated from light, to then make this sugar. So here is an overview of the whole reaction. We're going to use 3 CO2s and 3 ribulose 1,5-bisphosphates. Remember, those are our carrier molecules, ribulose 1,5-bisphosphate, and ultimately generate 6 3-phosphoglycerate. So 6 molecules of 3-phosphoglycerate. Okay, this is the first step that we go through. This 3-phosphoglycerate is then reduced into glyceraldehyde 3-phosphate, so 6 glyceraldehyde 3-phosphates. 5 of the 6 glyceraldehyde 3-phosphates are actually used to regenerate that ribulose 1,5-bisphosphate that we need in the beginning of the cycle. So ultimately, we only get out 1 glyceraldehyde 3-phosphate as our product. When we look at the net reaction of what is happening without thinking of things being regenerated, et cetera, when we cancel everything out, we just look at exactly what is put in and what is produced in the net. We put in 3 molecules of CO2, 3 carbons. We have to use 6 NADPHs. So 6 NADPHs have to get used and 9 ATPs have to get used. 3, 6, 9. 3 CO2s, 6 NADPHs, 9 ATPs. And what we make is a glyceraldehyde 3-phosphate. That's a 3-carbon molecule. That's where our CO2s went. We use up our NADPH into NADP, and we use up our ATP into ADP. Here is the actual process of the Calvin cycle outlined below. Stage 1 is what's called the fixation stage. This is where we're going to actually take incoming CO2 and bind it essentially into molecules to then form our final products. You can see that this also requires ribulose 1,5-bisphosphate. That is our carrier molecule. So we're going to take ribulose 1,5-bisphosphate and carbon dioxide, 3 molecules of each, and we're going to generate 6 of this molecule called 3-phosphoglycerate. This step, this fixation step, is catalyzed by an enzyme called Rubisco. That's its shorthand name. Rubisco actually stands for ribulose 1,5-bisphosphate carboxylase oxygenase. Again, shorthand, it's Rubisco. Rubisco is the enzyme that allows this first step to happen. It's the most important of the enzymes because it is the enzyme that actually captures CO2 and converts it into a carbon molecule, right? It's going to condense it with ribulose 1,5-bisphosphate and make these 3-phosphoglycerates, 6 molecules of them in total. It's a very large complex, just like its name suggests. It uses magnesium, and again, it's going to catalyze that first step of condensing our ribulose 1,5-bisphosphate with CO2 and making 3-phosphoglycerate. There's actually many forms of Rubisco because it is very important. It is the key molecule to this carbon capture and the assimilation of CO2 into making sugars. There is a Form 1 type Rubisco, which is found in plants and algae and cyanobacteria. And then there's also a Form 2 Rubisco, which is only found in photosynthetic bacteria, different than cyanobacteria. This particular enzyme actually has a low turnover number. This means that a lot of enzyme is needed. It doesn't act that fast. It's not as quick to do the reaction. So you have to have a lot of it around so that it can accomplish making sugars. For this very reason, 50% of all of the enzymes in plants, 50% are just Rubisco. It is so important, but also very slow. So plants have a lot of it around. Here's just a close-up of that Form 2 of Rubisco. You can see that it binds ribulose 1,5-bisphosphate via the magnesium. So magnesium is very important in this process, and it binds it in there. There's actually 2 per this Form 2 that bind into the pockets of the Rubisco. Here's a close-up of that actual center, the binding center. You can see the ribulose 1,5-bisphosphate, and here's our incoming CO2. Then there are some other amino acids part of the protein chain of Rubisco that are also interacting to hold that magnesium in place. But you can see that it's because of this magnesium coordinating the ribulose 1,5-bisphosphate and the CO2 next to each other that it's facilitating the reaction of them by just holding them close to each other. Rubisco is highly regulated. It is the first step of this assimilation of CO2. And of course, it is very important in the process of making sugars. So, this is the step that is regulated depending on plants' needs. Rubisco is essentially inactive until CO2 comes in and binds this portion of it, lysine 201, on the enzyme. There's also another enzyme called Rubisco activase, which can be triggered by light, that also helps to change Rubisco conformation to an active form. It also requires ATP. So if the plant is low on energy and doesn't have a lot of ATP around, it will not invest energy into making sugar. Okay, so that was stage one, again, the most heavily regulated and important stage of the Calvin cycle, which is using Rubisco to catalyze the condensation of CO2 and ribulose 1,5-bisphosphate into phosphoglycerate. The next stage is called the reduction stage, step 2. This is where we're actually taking that 3-phosphoglycerate and further reducing it through our NADPH to then form our final product, which is glyceraldehyde 3-phosphate. This is going to be a stage that uses ATP and NADPH to finalize the production of 6-glyceraldehyde 3-phosphates. One of those 6-glyceraldehyde 3-phosphates is going to exit the Calvin cycle at the end of this stage. The other 5-glyceraldehyde 3-phosphates are actually used with another set of 3 ATPs to then enter the last stage, which is regeneration. 5-glyceraldehyde 3-phosphates are needed to regenerate our 3 ribulose 1,5-bisphosphates. Remember that glyceraldehyde 3-phosphates are 3 carbon sugars, and ribulose 1,5-bisphosphate is a 5 carbon sugar. So we need 5 3 carbon sugars to make 3 5 carbon sugars. And that is the whole of the Calvin cycle. You can see that the Calvin cycle is pretty costly. Is very energetically costly. The fixation of 3 carbon dioxides just to form 1 glyceraldehyde 3-phosphate takes up 9 ATP molecules and 6 NADPH molecules. Remember that light reactions of photosynthesis produce ATP and NADPH at about the same ratio of 3:2. Once we do form glyceraldehyde 3-phosphate in a plant, it can then be further utilized for carbohydrate biosynthesis. Glyceraldehyde 3-phosphate is a known intermediate in glycolysis. It is an intermediate in gluconeogenesis, so it can be used to build glucose by going backwards. It can be broken down in glycolysis if you need things like pyruvate or oxaloacetate, which plants also do utilize those molecules. Don't think that plants won't use glycolysis because they don't need to then produce energy through glycolysis. They will still use glycolysis to make molecules like pyruvate to use in other processes. Gluconeogenesis, of course, is one of the main things we'll talk about because glyceraldehyde 3-phosphate can be shuttled into the chloroplast, and here it can be made into things like glucose, or it can be actually utilized to make starch, build up starch molecules from glucose and fructose. This is all going to occur mostly through the gluconeogenesis processes. Glyceraldehyde 3-phosphate can also be shuttled off into other areas to make things like sucrose. So the photosynthesis of one molecule of glyceraldehyde 3-phosphate actually requires the capture of about 24 protons. And this is from the proton movement from the stroba to the cycloid. That is where that is coming from, that number is coming from. Now, we talked about the ATP formation, but the ATP formation, or ATP used from the hydrolysis of stage 2 of the Calvin cycle, 8 of the 9 phosphates that are occurring during that portion can combine with ADP back in the stroma to regenerate ATP. But the 9th phosphate is actually incorporated into the glyceraldehyde 3-phosphate. It has the word phosphate in that. This process requires phosphate to actually be transferred from the cytosol into the inner area, and it uses a special port to do this. This is called the phosphate, inorganic phosphate, triose phosphate antiporter. And you can see it allows for the movement between the stroma and the cytosol. So again, this is the inner plant cell, the inner chloroplast stroma, and the cytosol area of the chloroplast. It allows transfer of sugar molecules. So glyceraldehyde 3-phosphate can move between the two. And it also allows for the movement of inorganic phosphate between the two to use in the production of glyceraldehyde 3-phosphate. So you can see here we have our movement of these products. Glyceraldehyde 3-phosphate can actually be transformed into dihydroxyphosphate, which can move through from the stroma to the cytosol. So here's our glyceraldehyde 3-phosphate, be transformed into dihydroxyacetone phosphate, moved out into the cytosol where we can remake it into glyceraldehyde 3-phosphate. And during this process, it is an antiporter. So in the opposite direction, we're moving an inorganic phosphate in. That inorganic phosphate, of course, can then be used further to make more glyceraldehyde 3-phosphate. This antiporter is important, obviously, for the movement of sugars and inorganic phosphate, but it's also needed to synthesize sucrose. So unlike big molecules like starch, which are just made in the chloroplast stroma, sucrose is made in the cytosol. So there is a need to transport our glyceraldehyde 3-phosphate precursor out into the cytosol or to other plant tissues via this antiporter. Remember that the inner chloroplast membrane, just like the inner mitochondria membrane, is impermeable to phosphorylated compounds, so you have to move through these transporters. So all in all, we're moving from light to CO2, ultimately to glyceraldehyde 3-phosphate within this plant. The photosynthesis of one glyceraldehyde 3-phosphate molecule is accompanied by magnesium transport from the cycloid to the stroma. This is because of the need for magnesium, if you remember, in our molecule rubisco. So enzyme for photosynthesis and CO2 assimilation, both in those processes, we use magnesium, and they have to occur in an alkaline environment. Thus, high magnesium conditions of the stroma. Here you can see a nice outline of everything that's occurring. This actually shows you the compartments of the cell too, to get a better idea. Here is the actual stroma of our chloroplast, and here is that inner thylakoid. The inner thylakoid is where that photosynthetic process is occurring inside the stroma of the chloroplast. Remember that this is where light is then converted eventually into our products through the electron transport chain to make ATP and NADPH. The ATP and NADPH is made in the stroma itself, where the products are then used for CO2 assimilation through our Calvin cycle to make glyceraldehyde 3-phosphate. Now remember, this is still inside the stroma. There is still the outer chloroplast, which is out here. So we're actually talking about kind of 3 segmented areas. That glyceraldehyde 3-phosphate can then be shuttled out to the chloroplast, where it can be used to make sucrose, for example, or it can stay in the stroma and be used to make glucose or to make products like glycogen or starch, more specifically. You can see that magnesium is actually inside the thylakoid, but can be transferred out into the stroma for its use in CO2 assimilation and actually producing glyceraldehyde 3-phosphate. So during the formation, during light generation and formation of our products, ATP and NADPH, we're also moving magnesium out into the stroma. And I want to remind you again, opposite to what you see in our mitochondria, this inner portion is where we have all of the H+. This is where we have the buildup of protons. This is very acidic. The outer portion here is not very acidic, and it is more basic in its pH. Here's just an example of the activity of a particular enzyme. This is fructose 1,6-bisphosphate in different pHs and with different concentrations of magnesium. So you can see that combinations of high pH, so more basic, and high magnesium allows the functionality of this particular enzyme. This is involved in glycolysis. Remember it from glycolysis and gluconeogenesis. Now, this process of having a basic and having lots of magnesium is going to occur because of the thylakoid intaking light pumping protons to the inside, making the stroma basic, and pumping magnesium into the stroma. So the process of actual light activation is what makes sure that the stroma has the correct conditions to ultimately allow for processes to occur, including CO2 assimilation and further processes of utilizing glyceraldehyde 3 phosphate in production of sugars. So light, in a way, is an activator of the whole process within the stroma, because light is going to make it so that the conditions are correct for certain enzymes to act. Light can also more actively activate, it can directly activate, I guess, enzymes to make them active. For example, ribulose 5-phosphate, fructose 1,6 bisphosphate, pseudoheptulose 1,7 bisphosphate, and glyceraldehyde 3-phosphate dehydrogenase. All of them are target enzymes for light activation. In this case, enzymes are inactive if oxidized. So if they are oxidized, that means cysteines have a disulfide bond, the enzymes are inactive. However, in light, photosystem 1, if you remember, the last stage, sends electrons to ferroredoxin. Ferroredoxin can send these electrons to thioredoxin, which ultimately breaks these bonds, activating these enzymes. So these enzymes are partially activated in some manner through a series of steps, ultimately because of light. Light has to hit to make those electrons, to get the electrons to ferredoxin, to then break those cysteine bonds and make these enzymes active. Here's just kind of a cartoon of what's happening. Light goes to photosystem 1, ultimately moving electrons to ferroredoxin. Ferroredoxin then moves these electrons to thioredoxin, which then is used to actually break disulfide bonds on enzymes, activating the enzymes. Now, we said that our triose phosphates, like glyceraldehyde 3 phosphate, of course, can be converted to larger molecules besides glucose, like starch and sucrose. Starch is made inside the chloroplast in the same area that we're able to make things like glucose. It's also made in amyloplasts and other plastids in certain plants. So things like tubers, like potatoes, certain seeds, certain roots that need a lot of starch for long-term storage and use. Um, these are going to have areas where they make and store starch as well. And they're going to provide bulk energy that is stored up in plants to use later. Plants, just like animals, can take that starch and re-break it down if needed. If there's a case where there's no light, plants can act in the reverse. Sucrose, again, is made in the cytosol, and it's usually used just to transport around the plant. So if sugar is needed in other parts of the plant, sucrose is kind of moved around to those areas out in the cytosol to then be broken down and utilized. Sucrose is kind of like the blood glucose of the plant. Instead of having glucose as that transport molecule and signaling molecule that moves around, signaling and is utilized in other areas, plants use sucrose, which is nice for us because sucrose is tasty. Again, we noted that many of these enzymes are regulated by light and dark. This is important because If it is dark, there is no fuel for the Calvin cycle. Calvin cycle is what's used to build up these sugar molecules. But if there is no light making NADPH and making ATP, we cannot make uh, we cannot continuously fuel this very expensive Calvin cycle to continuously make glyceraldehyde 3 phosphate. So you can't make sucrose. But in these cases, sucrose is instead needed so that it can be broken down in the dark to use for energy. So certain enzymes like fructose bisphosphotase 1. One second, sorry. So again, enzymes that are part of things like gluconeogenesis and glycolysis are going to be changed in their activity to regulate when plants are essentially breaking down sugars or making sugars. So for example, fructose bisphosphotase 1 is inhibited because it is part of gluconeogenesis, and we don't want to be making sugar in the dark. Phosphofructokinase 1 is active. This is part of something like glycolysis in plants. So in the dark, it's going to be breaking down sugars. Again, in the light, these are reversed, right? We're going to be making sugars and activating gluconeogenesis in the light, but in the dark, we're going to be inhibiting glycolysis. So gluconeogenesis of 2 of the glyceraldehyde 3 phosphates ultimately can, of course, result in glucose 6 phosphate, which is then used to make starch. And this is through phosphoglucomutase, which converts glucose 6 phosphate into glucose 1 phosphate, ultimately, which can then be used to build our starch molecules. Starch is also regulated in plants, again, via light. So this particular enzyme I just noted is going to be activated or deactivated whether you have a light, bright light, or a dark situation, or even in dim light, it's differently regulated. So the process of taking this glucose 1 phosphate and adding an ATP to it allows for the building of ADP glucose and ultimately building up our glycogen, excuse me, starch. I keep saying glycogen. So using ATP to build the ADP glucose and then to ultimately make our starch is going to be regulated via this light or dark. So you can see that in darkness, ADP, if there's a lot of ADP because it's dark, it's going to inhibit this process of the ADP glucose pyrophosphorylase. But when we have bright light and we're producing 3-phosphoglycerate via photosynthesis, this will activate this process. This is the rate limiting step in the actual production of starch. And again, is regulated ultimately by light. Okay, well, that is it for our biosynthesis of carbohydrates. We went through a lot about plants and looking at both how they intake energy from our into the chloroplast in the cycloids, taking in light and generating products like NADPH and ATP, and then how they use those in the Calvin cycle to produce precursors, specifically glyceraldehyde 3 phosphate, to then build up things like glucose, sucrose, or starch. Additionally, we looked at a lot of activation. So we looked at how light can regulate in different ways whether a plant is using its resources to make sugars or it is instead breaking down sugars to then use as energy.